The homocysteine-methionine dance. Homocysteine, an amino acid, was discovered in the early 1930s by two scientists, Vincent du Vigneaud and Robert J. Williams. Vincent du Vigneaud, an American biochemist, was awarded the Nobel Prize in Chemistry in 1955, for his work on synthesizing and characterizing various sulfur compounds, including homocysteine. He identified and named homocysteine in 1932 while studying the metabolism of sulfur-containing amino acids. Robert J. Williams, another American biochemist, independently discovered homocysteine around the same time. Their research laid the foundation for understanding the role of homocysteine in various physiological processes, and its association with health conditions such as cardiovascular disease. Vincent du Vigneaud and Robert J. Williams contributed significantly to our understanding of homocysteine and its role in physiological processes. They discovered that homocysteine is an intermediate compound, formed by the metabolization of the essential amino acid methionine through various biochemical processes. They also identified the pathways through which homocysteine can be converted into other metabolites, such as cysteine, or back to methionine. Additionally, their research demonstrated that abnormal levels of homocysteine in the body are associated with several health conditions, particularly cardiovascular disease. High levels of homocysteine are believed to contribute to the development of atherosclerosis, blood clot formation, and damage to the blood vessel walls. Their findings highlighted the importance of homocysteine as a potential biomarker and target for intervention, to reduce the risk of cardiovascular diseases. Following their initial discoveries, Vincent du Vigneaud and Robert J. Williams continued to contribute to the field of homocysteine research, making further discoveries that confirmed and expanded upon their initial findings. They conducted extensive studies on the metabolism and biochemistry of homocysteine, elucidating the enzymatic pathways involved in its conversion to other compounds. Du Vigneaud's research mainly focused on the synthesis and purification of homocysteine, and the identification of its chemical structure. Furthermore, both scientists continued to explore the association between homocysteine and cardiovascular disease. Du Vigneaud's research contributed to understanding how elevated homocysteine levels may promote atherosclerosis and clot formation. On the other hand, Williams conducted studies on homocystinuria, a genetic disorder characterized by high homocysteine levels. His investigations revealed the link between homocystinuria and severe cardiovascular complications, further supporting the connection between homocysteine and heart disease. Now let's begin. Inside every one of our body cells exists a crucial process called methylation. This process plays a vital role in maintaining good health and overall well-being. Imagine it as a delicate dance where a tiny group, called a methyl group, gracefully moves from one molecule to another, affecting numerous biological processes. One of the most fascinating aspects of methylation is its profound influence on our genes through a process known as DNA methylation. It's like a master switch that controls the expression of our genes. DNA methylation is a process where genes are likened to being snugly wrapped in a cozy blanket. It regulates gene expression by turning certain genes off and maintaining their silence. DNA methylation plays a crucial role in shaping cellular function and maintaining cell identity. In contrast, genes that remain unmethylated are active and unrestricted in their expression. This can potentially lead to detrimental effects on biological processes and contribute to the development of disease states. This ability to methylate our genes is of critical importance. It allows us to choose which genes should be expressed, like selecting the perfect playlist for different situations. When genes are methylated, the primary function is to regulate gene expression by turning them off and keeping them in a silent state. Methylation typically occurs in specific regions of DNA, such as promoter regions. It acts as a mechanism to inhibit gene transcription. By adding a methyl group to the DNA molecule, methylation can prevent the binding of transcription factors and other proteins necessary for gene activation, effectively silencing gene expression. However, it's important to note that DNA methylation is a complex process, and its impact on gene expression can vary, depending on the specific gene and context. Methylation patterns can be dynamic and change in response to various environmental factors and developmental stages. Sometimes, methylation can also be associated with gene activation or other regulatory functions. The overall effect of DNA methylation on gene expression is a finely tuned interplay between various regulatory mechanisms in the cell. Various factors influence our genes and their methylation. Some are inherited from our ancestors, while others are shaped by our environment and our lifestyle choices. Changes in methylation could have significant impacts on our health, leading to the emergence of diseases such as cancer, heart problems, and neurological disorders. Now let's focus on the central character, homocysteine. Homocysteine is an amino acid, and its conversion through methylation is crucial for the production of other important compounds, such as methionine and cysteine. If homocysteine levels are allowed to rise unchecked, it could spell trouble for our health. It's like a villain lurking in the shadows, waiting to wreak havoc. Elevated homocysteine levels are linked with increased risks of cardiovascular problems, such as heart disease, strokes, and blood clots. It could also affect our neurological function, potentially contributing to conditions such as Alzheimer's disease, dementia, and cognitive decline. It's very clear that this villain has to be dealt with. Fortunately, our bodies have a plan. They rely on specific nutrients to transform homocysteine into a hero called methionine. This conversion happens through two pathways, the long and the short paths. In the long route, B12, folate, and SAM are the superheroes that facilitate the transformation. TMG, trimethylglycine, and SAM, S-adenosylmethionine, are like superheroes who come to the rescue in the short pathway when it's necessary. It's important to note that this shortcut is limited to the liver and kidneys. Together, they prevent the dangerous accumulation of homocysteine, and ensure that our bodies function optimally. The shorter pathway involving TMG and SAM is primarily limited to the liver and kidneys, due to the specific enzymes and metabolic processes involved. These organs have the necessary machinery and enzymes to efficiently utilize TMG and SAM in biochemical reactions. The liver, being a central organ for metabolism and detoxification, plays a crucial role in various metabolic pathways, including the processing of homocysteine through TMG and SAM. The kidneys also contribute to the metabolism and elimination of waste products, including the breakdown of homocysteine. Therefore, while TMG and SAM have their superhero-like role in the body, their actions are mainly concentrated in the liver and kidneys, where the required enzymes and metabolic pathways are readily available. Methionine, the superstar of these conversions, plays a significant role in various bodily functions. Methionine is like a versatile building block, involved in synthesizing proteins, producing DNA, and carrying out other vital tasks. Additionally, methionine plays a vital role in our bodies by generating SAM, which acts as a catalyst for numerous methylation reactions. SAM serves as our primary methyl donor. The conversion of homocysteine has a third pathway. This pathway is necessary for producing antioxidants and other vital compounds. With the help of vitamin B6, homocysteine is further metabolized to form cystathionine, which is then transformed into cysteine. These are essential amino acids, playing their unique roles in protein synthesis and the production of antioxidants, such as glutathione. Vitamin B6 is the guiding force behind this transformation. However, the significance of methylation doesn't stop there. It impacts various aspects of cellular functions, including gene expression, neurotransmitter synthesis, detoxification processes, and metabolism. It's like a conductor orchestrating the harmony of our bodies. We support the methylation pathways by ensuring we consume adequate nutrients such as B12, folate, TMG, B6, and SAM. This, in turn, allows our cells to carry out essential processes and promote optimal health. In this grand tale of methylation, we realize that maintaining a delicate balance is crucial. We can shape our genetic expression and influence our well-being through proper methylation. By adopting a well-rounded diet and a lifestyle that promotes methylation, we can protect our health and mitigate the potential risks associated with elevated homocysteine levels. Thanks for watching.